Hi, everybody. Welcome back. Today we are going to discuss Chapter 4 of the Snustad Genetics textbook, which is entitled Extensions of Mendelism. Now, if you remember in the previous work, we looked at Gregor Mendel's studies about the pea plant and established these important concepts of genetics, namely um, his idea that a single gene controls a single phenotype, that gene has two what we would now call alleles, and that those alleles are clearly dominant to one another. And we saw that Mendel had lots of success going through with color and shape and texture and all sorts of different phenotypes of the pea plant, and established some really important predictive tools for us for genetics. This chapter, I think, really just gets into this idea of all the other things, or many of the other things that we know are true in genetics that do not fit with Mendel's sort of simple, straightforward rules. Um, so uh, think of this one as a laundry list, maybe, as you're reading this chapter. As you go through the chapter itself, you're going to see a lot of terminology, like here, incomplete dominance and codominance are two distinct things that we're going to talk about. Those are two things that don't exactly follow Mendel's rules, of course. Mendel um, posited that one allele would be completely dominant over the other, and so we could have hidden phenotypes that would show up in subsequent generations. Um, all of the tools that we've learned in the previous chapters as far as predicting phenotype and genotype frequencies. So things like Punnett squares and probability calculations and forked line diagrams, all of those still work here. The only difference with these different systems is that the interpretation of a genotype to a phenotype is a little bit different. So with that, let's go ahead and look at our slides for this chapter. These are available on Google Slides, so you can check that out if you want to follow along in the text. But here's our list of things that are talked about in this chapter. So we're going to discuss incomplete and codominance to start. And then we'll discuss this concept of multiple alleles and what that looks like when we have more than two alleles, as Mendel sort of started us out with. Then we'll talk about some vocabulary and types of mutants and their manifestation. And we'll talk a little bit at the end about inbreeding and hybrid vigor. So here we go. Incomplete dominance is this first trait, or not trait, that's not right. It's the first sort of pattern of inheritance we're going to talk about. And the example given in the text is of a snapdragon. We learned from Mendel's work that the pea plant has some colors to its flower as well. But in this case, the snapdragon flower can be observed to have three different colors. And importantly, that pink color is an intermediate phenotype. And so when we talk about incomplete dominance, we're always going to be looking at extreme phenotypes, in this case, white and red, and then another phenotype that's intermediate. And so we can see here in the second column what the genotypes would look like. So using the same kind of lettering that Mendel would have used, or that we use when discussing Mendel's work. We have the capital letter here for the dominant, in this case, a red pigment producing gene. When we have that in its homozygous form, we see a lot of that pigment, and so the flower looks red. When we don't have that at all here, our homozygous recessive, we see the absence of pigment. We see a white flower. But now the difference is in the heterozygous case. Now, if this followed Mendelian genetics, these heterozygotes would be red. We'd have that dominant W allele here, masking the presence of the recessive lowercase w. This would look red. In this particular case, we see that it in fact looks pink, and that intermediate phenotype is the key here. So we would say that the dominant capital W allele is incompletely dominant over the recessive lowercase w. This should not be confused with the next concept, which is called codominance. Codominance is one in which we have more than one dominant allele that could both be presented. One effectively doesn't change the impact of the other, so we can have two different things that both are presented, and in that case, we would say these are codominant. Now, I don't really love this example. I'm using this example because it is the one that's presented in the textbook, or one of the ones that's presented in the textbook, the MN blood typing antigen system. What they're showing us here is that there are two dominant alleles. Now, I wanted to point out that in the terms of the genotyping, quite often what we'll do now is we'll use a capital or a lowercase letter to represent the allele itself, in this case the L. That's a capital L because this is the dominant allele. And then we'll use a superscript to indicate which dominant allele we're talking about. So in this case, we have a situation where we're homozygous for the dominant M allele. That would give us this blood type antigen being produced. That is the M type antigen. And so we would see a reaction with anti-M serum. This is a blood typing thing. So effectively, we would say this sample is type M. Similarly, if we have homozygous for the dominant N allele, we would only produce the N antigen, and we would see blood type N in this petri dish over here. This clustering is how we know something is positive for blood typing. That's not really relevant to us, but that's what we're seeing. But in the middle, again, it's the heterozygote that's interesting. So when we have the dominant M and the dominant N alleles both present, we in fact produce both antigens, the M and the N, and this could be producing a protein or a pigment or a carbohydrate structure on the surface of a cell, something like that. Whatever it is, we're producing some biochemical thing. And in this case, both of those things are present. So the M and the N don't influence one another. This is not incomplete dominance, where the thing we get is like a blended M-N kind of a thing. This is two separate, distinct things both being produced. What I don't love about this picture is that it sort of forgets for us that there could be recessive alleles as well. So there could be more than just these two. There could be a lowercase l that's recessive to both, for instance. And we actually see that in the other blood typing example in the book, which is the maybe more familiar to you, ABO blood typing. In that case, the A and the B alleles are both dominant. They're codominant, and they are dominant to the recessive, what's listed as a lowercase I in that system, but the recessive O type. And so look at that example as well. That's a 3-allele system that has a pair of codominant alleles and one recessive allele. Another example here, and this could be the human ABO blood type as well, multiple alleles of a single gene. I just mentioned that the ABO blood typing system has 3 alleles, a pair of codominant alleles and a recessive. This example shows us a series of bunnies. Now you're going to see lots and lots and lots of examples in the textbook about bunnies. If you go through the problems at the end of the book and the worksheets online and things like that. Don't stress about trying to sort of memorize what these mean, okay? Treat this slide, this diagram out of the chapter, as your Rosetta Stone. All right, this is telling us that there are 4 different alleles in this system. We have simply the recessive C allele here. I hate using W's and C's because they're hard to write to make a difference between lowercase and uppercase. But fortunately for us, in the rabbit system, the sort of wild type allele is listed here with a plus. These are all listed with superscripts. And so when we have the plus plus allele system, we see what's called a wild type rabbit. Now, wild type in genetics terminology doesn't necessarily mean that it's the most dominant allele. It just means that it's the most common allele that you'll find in a population. That probably means that there's some dominance, but if a different sort of more dominant allele were to be introduced, it could potentially take over and become wild type later on. For our purposes, we have 4 alleles. We have the C plus wild type allele. When that is homozygous, we have this sort of brownish looking kind of normal field bunny. If we have the recessive C with no superscript, if that's homozygous, we see an albino bunny. And then there are 2 other alleles they're telling us about. So the bunny could have this C with the H superscript. This is the so-called Himalayan allele. A bunny that has that in its homozygous genotype, as shown here, would be basically albino in the body, but it would have these little black sort of tips, paws, nose, tail a little bit, and ears. This is just what the Himalayan bunny looks like when it's in the homozygous, when it has 2 of these alleles. And then there's a 4th one, the CH allele, the chinchilla allele. This bunny looks gray in the picture, and I guess it is, but what it really is white hair that has the tip of the hairs are dark. And so it's not like the Himalayan where it's white and black in different regions. Each hair has a white and a black portion to it. Okay, so here's our Rosetta Stone. There are 4 alleles in this system. This is what they look like when they're in their homozygous state. Now, maybe you've asked here, what happens if we mix them? What if I had a heterozygote of any of these pairs? Well, that's what our next diagram shows us. And this is establishing that series of dominance, okay? So what we see here is a really complicated system. And basically, if we start from the top, if we have the presence of at least one of the C plus wild type alleles, our bunny looks wild type. So this is, sort of classic dominant recessive behavior. The C plus is dominant to all 3 of these other ones. And so whether we have a homozygous C plus or we have a heterozygote that has C plus and anything else, we get the same look. Okay, that's clear dominance. Down at the bottom, we have another example of clear dominance. That if we have the Himalayan allele paired with the recessive albino allele, the Himalayan allele is dominant. The bunny looks Himalayan. So clear dominance at the top, clear dominance at the bottom. Now what's happening in the middle? Remember, if we had the homozygous chinchilla bunny, let's go back and look. The homozygous chinchilla bunny would look sort of this dark gray color. When we see chinchilla heterozygous with the recessive albino allele, we see a lighter color. So this is sort of that incomplete dominance, right? We see this intermediate phenotype. It's not the gray of the chinchilla bunny. It's not the white of the albino. It's somewhere in between. So we could say with some certainty that the chinchilla and the albino alleles here are incompletely dominant to one another. Likewise, down here, the Himalayan and chinchilla alleles, when we see those, we don't see just the chinchilla, we don't see just the Himalayan phenotype, we see this sort of intermediate bunny phenotype. A little bit darker fur, still has the black tips. And so this is a very complicated system. Again, don't memorize all of this, right? The point is to use these diagrams as your guide. And then when you do problems in the book or problems on the worksheets, you'll be asked to do crosses between certain bunnies with certain genotypes. Do whatever technique worked for you before. Use the Punnett square or use probabilities or whatever it is. You'll get your genotypes listed out and then use this diagram and this diagram to assign those genotypes to phenotypes. Okay, if you see this this heterozygote, chinchilla and albino in one of your boxes of your Punnett square, you're just going to say, well, that means it's light chinchilla. Okay, you don't need to memorize it. I've put a couple of words here on the side just as a sort of a sidebar vocabulary. In terms of genetics, we mentioned wild type is the allele that represents the most common allele in a population. It tends to be dominant over the other alleles in the population. It doesn't have to be, but it tends to be. A null allele, excuse me, is one that would be non-functional. So whatever its function is, it doesn't do that thing anymore. We could maybe say that the albino allele here is non-functional. It's a null allele. some mutation has occurred in that gene sequence that makes it not do whatever it's going to do. Typically, or oftentimes, we would talk about a structural function being broken, or we would talk about a an enzyme that has some biochemical function not accomplishing its goal anymore. The other kind of allele we can have is a hypomorphic allele. These are very common. This is a partial loss of function. So it still works, kind of, but it doesn't work as well. And maybe we could say that about the chinchilla and Himalayan, although I'm not sure that word necessarily applies. But this would be a case where, maybe some enzyme is supposed to process material at some certain rate, and some mutant does it at a lower rate, for instance. That would be considered a hypomorphic allele. If the enzyme just doesn't work, it doesn't process substrate, that would be a null allele. And so we can see kind of some of that in this diagram from the text, that if we imagine a piece of DNA, a gene, having some certain allele here, that through the miracle of cellular mechanics produces a functional polypeptide. Now this thing could be structural, it could be functional, whatever it does that contributes to a wild type phenotype. Now, an amorphic or null allele is one in which that particular genetic sequence produces some messed up version of that polypeptide. So it doesn't accomplish the structural goal it was supposed to, or it doesn't have the same enzyme binding sites it was supposed to in the wild type state. And so it has a mutant phenotype. Now this has severe mutant phenotype because a null allele is broken. A lot of times this happens when we have a mutation that introduces a stop codon. So remember in transcription, when you go through, you could have RNA termination happen early, or in translation, if you happen to introduce a stop codon, you would have translation stop at that point. If we stop partway through a protein, I hope it makes sense that that protein is probably going to be pretty messed up. The one below that, a recessive hypomorph, a loss of function or reduced function allele. This is one in which we sort of get a normal structure, but something's a little messed up. And so we get some variant of a mutant phenotype. And there could be tons of these. The more that we understand genetics, the more we're able to find these alleles. When we generate these genomic databases, like the Human Genome Project and all the other genomes that are posted on the government's websites and private industry's websites and things, we take some certain genotype, some certain gene sequence to be normal, and we compare everything else to that. So we have the reference sequence, and then everything else is compared to that. In some cases, maybe a phenotype literally doesn't change the amino acid sequence. That would still give us wild type. That's that idea of a silent mutation that can occur. However, in many cases, we can have amino acid substitutions that lead to different degrees of mutant phenotype or reduced function. Those are very, very common. In many cases, we can end up with something that's severe or where the protein has been completely dysfunctional. The one at the bottom is kind of interesting, and this is a dominant negative. Dominant negative is a concept where some mutation in the gene creates some different polypeptide than normal. But the key thing here is not just that that polypeptide doesn't work. Okay, maybe it doesn't work, but the key thing is that that polypeptide interferes with the wild type. So this mutation ends up being a severe phenotype because not only are you producing this messed up protein, but that messed up protein also shuts down or interferes with the function of the normal stuff. And so if you had a dominant negative that was in a heterozygous genotype with the wild type, the dominant negative would effectively mask the wild type. And that's why it's called a dominant negative. It has a negative effect, but it genetically would act in a dominant way. We've talked all about these different alleles. In today's modern age, one of the ways that we could assess this is just to sequence everything, right? Why not just sequence everything? sequencing is expensive. Today it's a lot cheaper than it used to be, but it's still expensive. And I don't know about you, but I don't necessarily keep a DNA sequencer in my lab. I would love that, but oftentimes we send our sequencing projects away to core facilities, either at our own institution or somewhere else. Um, but one way that we can test for allelism is to um, create new strains, to mate strains together with what are called tester strains. And this is super common. I did this all the time in the East Lab when I worked there. We would have all these different genotype strains that we knew were defective in various different genes, and we would cross our new mutation, strain with a new mutation, with each one of those. And the idea here is complementarity. So if we cross a strain that had a messed up C gene with a strain that had a messed up A gene. Okay, now remember, we're presuming that all the other stuff is normal. So the A strain here would have a normal B and a normal C and a normal D, whatever. If we did that, the broken C alleles from this new strain would be complemented by the good C alleles from the tester strain. And likewise, the messed up A alleles here would be complemented by the good A alleles in our mutant. And so we would get wild type. That hybrid phenotype would tell us that those two mutations are not in the same gene. So I could do that with a whole bunch of these. And literally you would do this on a plate. You'd have a whole bunch of, well, I guess depending on the organism, if it's yeast or bacteria, you would do it on a plate. You'd have a whole bunch of little spots on your plate, little grid that you'd be working on. And in this case, what I would find out is that, oh, look, when I cross my new mutant that I don't know what gene is mutated, I cross it with my C tester genotype that has mutant C alleles, I get a mutant phenotype. That tells me that my original mutation that I was dealing with, that I was testing here, my brand new thing, is in the C gene because I can't complement it with the C mutant strain. So that tells me that my new thing and this old thing have maybe the same mutation, I don't know, but they're in the same gene. They are alleles of one another. And there are established strains, tester strains that people use in the different fields, whether you're talking about E. coli or yeast or worms or flies or whatever. Here's an example of that in fruit flies. Now, fruit flies have red eyes, typically. The wild type is sort of this, from this picture, maybe you can tell, I don't know, this sort of dark reddish kind of eye. But there are many mutations that influence the color of the fly's eye. They make it brighter, or they make the eye itself larger or smaller. In this case, these are 3 different mutations that have been shown to cause this sort of bright red eye phenotype. In the fly, their mutants are given cool names. So the first one here was called Cinnabar, and then there was another one that was called Cinnabar 2 that maybe gives something away. But there's a third allele called Scarlet that looks very similar. Turns out, if you do this sort of crossing, if you cross a Cinnabar fly with a Scarlet fly, you get wild type eyes again in the hybrid. So this tells us that Cinnabar and Scarlet are mutations in 2 different genes. The good copy of Scarlet in the Cinnabar mutant and the good copy of Cinnabar in the Scarlet mutant complement each other, and they produce a normal fly. Same thing happens in the bottom line down here. But why are Cinnabar and Cinnabar 2 called Cinnabar and Cinnabar 2? Well, it's because we now know from this kind of a test that they are in fact 2 different alleles of the same gene. When we cross them together, flies that have this Cinnabar 1 maybe mutation with the Cinnabar 2 mutation, we still get a mutant phenotype. This tells us that they're in the same gene. They're alleles of the same gene because the Cinnabar gene is messed up here. It's also maybe differently messed up here, but when I cross these together, I don't get a normal Cinnabar eye formation gene. I don't get a wild type. By doing this, we can come up with all kinds of different mutations. Now, these are phenotypic mutations here, so they're not, we don't need to sequence. We can just do crosses. What we can do with careful analysis is really map out a pathway. So we could say, for instance, here that the cinnabar gene and the scarlet gene are both involved in forming the proper eye color. Now, what they do, we don't know from this exact experiment, but they are probably both involved in mapping out the eye's color, but we know that they're in different genes. So maybe they're sequential steps in a pathway. Maybe they encode two different pieces of some molecular machinery that does something important. We don't exactly know, but we do know that genetically they're in two different genes. This is very, very easy to do in bulk. You can cross, hundreds and hundreds of samples pretty easily and much less expensive than trying to sequence everything. Much faster, actually. So what we've talked about so far are mutations that have obvious changes of phenotype. So eye colors are different, flower colors are different. Blood cells produce certain structures that we can test with antibodies, things like that. Those are visible mutations. I mentioned in passing silent mutations. These would be the ones that don't change our amino acid content. So remember, our genetic code is wobbly and redundant. So if we had a mutation that changed us from one phenylalanine codon to another, that's a silent mutation. Doesn't make any difference. The gene sequence is different technically, but the amino acid sequence is not. Those kinds of mutations can have an impact if they happen to fall in a control region or a splicing region. We'll talk about some of that molecular stuff in the second half of the class. But for the purposes of phenotypic genetics like we're doing now, you wouldn't see those. We do have lethal mutations as well. Now, lethal mutations are more evident in the numbers. If your numbers don't quite work the way you expect them to based on your Punnett squares or your statistics, it might suggest the presence of a lethal mutation. In this case, that's where individuals that are homozygous for this particular gene sequence don't survive. So it's not that you can count them, right? They don't survive. So if we're talking about animals, they tend not to be born in the first place. If we're talking about plants, you often wouldn't see these seeds germinating. The seeds would not make it into a functional plant. So these are sort of invisible. And so the way that we find these is to look for altered statistics. The other category would be a sterile mutation. So this is an individual that is functional. It does its normal, animal or plant things, but it can't reproduce. So there's some defect in usually in gamete formation, whether we're talking about pollen and egg in plants or sperm and egg in animals. Or in cell division, if we're talking about some other organism that reproduces by fission or something like that, there's some problem that results in not reproducing. So here's an example of that lethal mutant that we just talked about. This is using mice, and we can see now in this example, we have a couple of dominant alleles. So it should be a codominant kind of a situation. And at the top, what we see is that if we take two yellow mice, now that yellow is encoded by that capital Y superscript, sorry. And we see both of these mice are genotypically heterozygotes here. We have the Y yellow allele paired with the wild type allele. If we cross them, we make our little Punnett square, right? So we say, okay, we've got one of two choices from the one parent and one of two choices from the other parent. We do our Punnett square thing. We see some of the mice are wild type, this word agouti. So that's our A plus A plus. We see some of the mice are just like the parents, that AY A plus heterozygote. And what I want to point out is that these AY AY ones down here, these ones wouldn't survive. So although it shows us a picture here, that's meant to be a little shriveled up embryo that didn't survive. Uh, remember, we wouldn't see those. We would mate these two mice together. We'd wait for the pups to be born. We would count the pups and um, assess them based on whatever phenotypes we're looking at. We would not say we have 1/4 of the offspring are wild type and 2/4 are yellow and 1/4 is dead because we never see those dead ones. So that's what I was talking about with altered statistics. What we would have in the actual generational offspring, the ones that we actually have born that we can count, are just these three categories. So we would see 1/3 of our offspring are wild type and 2/3 are yellow. And it's that presence of the thirds here, not fourths, that would tell us that something else happened, that there must have been a category that we didn't see. And embryonic lethality is the easiest way to explain that. In yeast, we know of a whole bunch of sterile mutants, whereas fruit flies get cool names for their mutations like cinnabar and scarlet and forked and bristled and all sorts of things. Yeast, we tend to name things in the yeast system with a three-letter abbreviation that typically has something to do with function, or at least what we think the function is. So the STE mutants in yeast are sterile mutants. These are mutations that were observed through those sorts of crosses and random mutagenesis and allele testing that we talked about a while ago. Um, they were all found to not allow yeast to reproduce. So yeast that was carrying a mutation in one of these STE genes could not reproduce. And so they're just numbered. The first one discovered was sterile 1, and then 2, 3, 4, so forth. There's a whole bunch of them. Um, and what we know now is that, uh, of course, their functions have been pretty thoroughly investigated. Sterile 2, for instance, is the receptor for the pheromone. Yeast come in two mating types called A and alpha, just to make it really terrible to type when you're writing a paper. Um, in order for those two cells to come together, much like a sperm and an egg, there's a pheromone um, sort of a signaling pathway that allows them to find each other. In the case of yeast, because they're non-motile, they grow toward one another based on pheromone concentrations. And so that pheromone that's secreted by the A cell has to be received on the alpha cell and vice versa. And then they grow towards one another. It's really a cool process. Turns out that sterile 2 is the receptor in the A cell that would have received the alpha pheromone. If we don't have that receptor, then the A cells don't realize they're supposed to be mating. They don't realize there's a mating partner next door, and so they don't mate. However, it turns out that 5 and 7 and 18, those mutations are in three different genes that are all components of the signaling cascade, that once that receptor is triggered, that signal has to get from the membrane where the receptor is laying all the way down into the cell to cause it to do something to start the mating process. Sterile 5, sterile 7, and sterile 18 are all different parts of that cascade process, that signaling process. So these are four different mutations that all have the phenotypic effect of being sterile, but for different reasons. So I'm not going to play this, but I would encourage you to watch Jurassic Park if you've never seen it before, or watch the new ones if you want to watch that. But go back to the old one. Jeff Goldblum here talks about life finding a way. And in the movie, if you'll indulge me for a second, the premise was that they recreated dinosaurs from some ancestral genome that they found in a bug, and the scientist told the visitors to the visitor center that, well, you don't have to worry about this because we've strictly controlled this, and these animals are all female and they can't reproduce. Um, and he goes on this sort of esoteric rant about how nature finds a way, and that turns out to be true. So in basically every system that's been studied, whether it's bacteria or yeast or worms or flies or whatever, we will have what are called suppressor mutations that occur spontaneously that tend to offset particularly deleterious mutations. So imagine population of cells that have this sterile mutation, right? Population of yeast cells, perhaps. They can't mate. Well, what if one of them has a mutation that compensates for that, that somehow fixes the deficit? That one then would survive where the other ones wouldn't. It has a selective advantage. The same thing is true with any stress that we put um, on our cells, on a biological system, that if we stress that thing, those that can survive and thrive beyond the stress will repopulate the system, I guess, repopulate and expand in number. That's a little silly. We'll skip that. Okay. So at this point, I like to sort of say, here's this laundry list, right? We've been going through. How lucky was Mendel? Now, um, if you go back and read Mendel's work and read some of the criticisms of it, this is sometimes a criticism that gets levied on him to say, well, how could it have been so perfect, right? How did he get so lucky? And, we can have an argument about that, whether he didn't talk about some of the things that were a little more confusing, which maybe is justifiable. Maybe he just did get lucky and picked a good organism. I don't know. But we've observed a whole bunch of different things. So in addition to the incomplete and codominance and allelic series and um, embryonic lethality that we talked about just now, we have this laundry list of other things. So environmental effects can have an impact on how genetics manifest themselves. We can have another term here called incomplete penetrance. Don't get this confused with incomplete dominance. I'll show you the difference. Variable expressivity, gene interactions in pleiotropy, and epistasis. So we're going to run through these here relatively quickly. I encourage you to look for all of these. Look at the problems at the end of the chapter. Really sort of look through those, practice, make sure that they make sense to you. Okay. Influence of the environment. So there's a whole bunch of examples. And basically, in the short version, is that genes can function differently or have a different impact on the organism depending on what's going on around the cell or around the organism. There are plenty of temperature-sensitive mutations in flies and worms and yeast and bacteria. These are mutations that maybe work perfectly fine at one temperature, but if you raise or lower the temperature a little bit, the protein stops working normally. Easiest way to think about this is with a protein that has a mechanical function. It's supposed to do something, that, it's supposed to bind something perhaps. And if you raise the temperature, the protein shape changes and it can't bind as well. That'd be one example. We have sex determination in certain animals here that is dependent on temperature. I like to think back to that Jurassic Park example. Don't worry, they're all female. Well, maybe not. Phenylketonuria is an example of a dietary problem. This is a mutation that can occur that has no problem if you don't have extreme amounts of phenylalanine in your diet. We'll see that in a minute. And then pattern baldness is a mutation that really impacts differently in different genders. And so here's that reptile thing, which I think is just super cool. This figure is taken from another textbook by Gilbert. So most reptiles use a chromosome-based sex determination like we do, right? So if you have certain chromosomes, you have a biological sex that gives rise to certain features. But most turtles have temperature-dependent sex determination. And what's shown here are three examples. Now we have a graph that shows the percent male on the y-axis. So if the line's at the top, that would be all male in the brood. And if it's at the bottom, it would be all female. And then we've got the temperature scale along the x-axis. So what we see here for this little red-orange turtle is that if we have a low temperature, we basically have all females. If we have a high temperature, we basically have all females. But if we have a temperature right in the middle, we get mostly males. And so this turtle would generate a reasonable breeding population if we were anywhere in this temperature range, right? We'd get a good mix of males and females in the next generation, and they would then be able to reproduce and continue. For this green turtle, the trend is a little different. Low temperature favors males, high temperature favors females, and that temperature range is a bit more narrow, where we'd get both. So just a few degrees. And so you could see where maybe a species like this might be particularly impacted by a slight change in temperature. If the ambient temperature were one season, a few degrees higher than normal, maybe you get mostly females in the next generation, and then that would impact the next generation because there wouldn't be very many males around to mate. See the same kind of trend with this alligator, the same kind of U-shaped trend we see for the red guy, but at a different temperature, right? So kind of a cool phenomenon. You can read more about it if you're interested. Here's PKU. PKU is a genetic disease. You may, if you're into drinking diet sodas, and you might recognize what this label is down here, but you'll see this little warning, and maybe you've seen it in the past and didn't know what it meant. This is an address to people as a group called phenylketonuria. Those are people with this disease, that the product contains phenylalanine. And the reason for that is that aspartame, which is the sweetener found in a lot of products like this, aspartame is a three-part little molecule here. A sort of orangey bit is a phenylalanine amino acid. And so when this hits your guts, it gets broken down into aspartate over here, phenylalanine in the orange, and a little bit of methanol here in the blue. Normally, for people who are wild type in all these genes, that phenylalanine that you consume would be modified by this enzyme into tyrosine, and then again into this product, and again, and again, and it would go on and on and on. There are two known genetic diseases mentioned here. People with phenylketonuria have a defect in this first enzyme shown, and people with alkaptonuria have a defect in the second enzyme. So in either case, you would not be able to conduct this pathway. People with PKU can't do this, so if they consume a lot of phenylalanine, they get a buildup of this in their bloodstream, in their system, which can have some effects. Again, if you don't consume large amounts of phenylalanine, then this mutation would go basically unnoticed. Now here's incomplete penetrance. We talked about incomplete dominance before. Incomplete dominance was that case like with the snapdragons, where we can have a blended phenotype for the heterozygote. Incomplete penetrance is different. And so what's being shown here is polydactyly, where we've got an additional digit on the hands. This is an extra thumb in both cases. In this case, what we're saying is individuals may not express a trait even though they have the appropriate genotype. And so when we look at this, pedigree, remember, if we see something expressed in every generation, we tend to think that's a dominant trait. And so we could pretty easily follow that trait from the first generation female matriarch to that second generation female to, say, this third generation female, and then down to a couple of the offspring. And so we could map this out, and we could come up with a pretty strong argument for this being a dominant allele. And maybe we could go further than that, but we can definitely say it's dominant. But when we look at the first family in generation 3 to 4 over here, we see that generation 4 in this first family has a third of the kids that have this trait. And if we're presuming it's dominant, that's fine, but then one of the parents should have it. And in fact, it should be the dad based on this family, and the dad doesn't have it. So how can we explain that? Well, this is incomplete penetrance. So what we would say is the genotype of the family, this whole pedigree, gives us a pretty solid determination that this thing is dominant, right? And it gives this phenotype. That tells us that this dad, individual 3-2, this individual must carry that mutant allele. But for whatever reason, we don't see the phenotype in that person. Now, why not? Who knows? It could be that it just didn't express properly. There could be an environmental concern to it. Could be another gene that's involved that we don't really understand. It could be something simple as not knowing, right? Maybe they just didn't notice. Maybe, and I mean, in this picture, how could you not notice this extra finger? But maybe it was more of a bump on this guy's finger instead of like a full-blown finger with a nail. Um, Maybe it was surgically removed as a baby, and those records are lost to time. We don't know. But this pedigree tells us that this is a dominant gene, dominant allele rather, and that individual 3-2 should have had it, must have had it in order to have affected children, but they don't seem to. That's incomplete penetrance. This is really common, especially as you go back in family records, right? The longer ago we're trying to look, the harder it is to find good records. Here's variable expressivity. Oh, I apologize for that. I don't know what happened to this diagram. Variable expressivity, this is a particular mutation in fruit flies. All of these little cartoon flies here all have the same exact genotype. Variable expressivity says the trait isn't expressed, the phenotype isn't uniform amongst individuals with the same genotype. So we have some flies here that have a basically normal eye. We have some where it's sort of the right size, but it's misshapen. We have some that are smaller, and we have this one that's totally absent. One gene having this same effect is unusual. We can just call it variable expressivity. In reality, further study usually indicates that there's something else happening. There's temperature sensitivity or there's another gene involved, and we just don't know about it yet. And then we come to chickens. Great example of gene interactions. So in case you've ever wondered, maybe you grow chickens yourself, I don't know, but the little wobbly bit here on the chicken that we see pictured is called a comb. And in this particular example, we have two genes that interact to form the shape of the comb. So again, treat this as a Rosetta Stone, okay, like the rabbits before. If we have a dominant R and a dominant P, both of those functions work. We produce this walnut-shaped comb shown in part C. If only R works and P is homozygous recessive, we get the rose comb, A. If only P works and R is recessive, we get the P comb, this little tiny guy here shown in B. And if both genes are homozygous recessive, we get what's called a single comb over here in D. Okay? So again, we can do a dihybrid cross, just like we would have with Mendelian genetics. Dihybrid cross of our parents, we can draw a Punnett square or probabilities or forked lines or whatever. We're going to get genotypes in each box like this. And then the difference here is that we have to use our knowledge now of these different phenotypes to interpret each box in the Punnett square. Okay. So when we look at this, we see a Wyandotte breed of chicken has a rose comb, which means it must have the dominant R and the recessive P. Now they're showing it as homozygous dominant because the Wyandotte is a true breeding strain. So just like Mendel's piece, this is true breeding, which means it's homozygous for whatever allele it's got. The Brahma chicken over here has a P comb, again, true breeding. Now we see when we do the gametes, there's only one choice, so we get hybrids of those. If we crossed a Wyandotte and a Brahma, all of our organisms would be heterozygous for both genes. That would give us that walnut comb based on the previous slide. And if we take two of our walnut chickens and mate them together, we would expect from a Punnett square 16 boxes here, right? Dihybrid cross. We're going to fill in all of our little phenotypes, right? And just like before, we would have said 9 out of 16 would be dominant phenotype. 3 out of 16 would be dominant for one, recessive for the other. And 1 out of 16 would be recessive for both. That's still true. The only difference here is the interpretation. So now we'll go in and say, oh, instead of saying like in the Mendelian case, maybe these are green and wrinkled or something. Now both of those genes contribute to the same phenotype. So we saw in the previous slide, dominant would give us walnut. Dominant R recessive P gives us rose. Dominant P recessive R gives us P, and recessive both gives us single. So it's just the interpretation part that's different. And use the diagrams in the chapter to help. Here's pleiotropy. Pleiotropy is a broad term that means that one gene can have multiple phenotypes. Oftentimes, this comes from metabolic pathways or longer, sort of bigger cellular machines. So PKU is one example, right? We saw this biochemical pathway here a few slides ago. Turns out if you have a mutation in phenylalanine hydroxylase, you have PKU. Those symptoms include mental impairment, lightened hair color, and the presence of altered metabolites in your blood and your urine. Why all of those things? Well, because phenylalanine is involved in many different cellular processes throughout the body. And so when we have that single gene mutation that blocks this pathway, we get increased levels of phenylalanine that then triggers increased or altered production in several different other biochemical pathways. Gets complicated. The same thing is true in Drosophila, and we've seen it in all kinds of species, but in fruit flies, the singed gene, which is so called because the bristles in these mutants, the little hairs on their legs, look like they've been burned. You know, if you've ever gotten too close to a campfire or something, you might see body hair get sort of twisted up. That's kind of what these look like just as a mutant. And so it does affect bristle shape. It also has an effect on their egg production, which is a totally different system, but that same gene influences both pathways. This is pleiotropy. So a single mutation causes multiple effects. And again, often it's because of these soluble precursors or links between different biochemical pathways. Epistasis is our last big topic for the chapter. Super important to my field of biochemistry and the way that biochemistry and genetics interact. This is where an allele of one gene can override the effect of other genes. So think about like a sequential pathway, for instance, a biochemical pathway, a transport pathway where step one has to occur before step two, protein modification, where I have to add one piece to the protein before I can then add another piece to it. Things like that, things that happen sequentially. The gene that does the masking is called the epistatic gene. So it's epistatic to something else. That's the way the terminology works. And we can look at this in terms of, pigment production, maybe at the top. Here's a linear biosynthetic pathway where we might start with some chemical that gets modified by the gene C in this example to make some intermediate. And then that thing gets modified by another gene to produce anthocyanin. This is that purple pigment in blueberries and snapdragons and stuff. So if we had C broken or P broken, either one, we would not produce our purple pigment, right? But what if we had them both broken? Well, if C was broken, our pathway would stop here. And so it doesn't actually matter whether P works or not in that case because our pathway is stuck back here. So we would say that gene C, or mutation in gene C, is epistatic to a mutation in gene P. Okay, we wouldn't have that same thing in the second pathway. If we have redundant paths, both gene products can convert precursor to product. Now I could break A and still be able to go through gene B function. I could break B and still be able to go through gene A, but if both of them were broken, it would stop the phenotypic production. This is not a case of epistasis. I don't have that one after the other sequential kind of an approach. Down at the bottom here is the same kind of thing we see up top, but now they're showing us that the dominant allele here blocks, that's what this T bar is, blocks this step. So recessive allows the step to go forward. It's a little bit different to what we see up top. But again, here's one where gene C is epistatic to gene G, right? If C blocks the pathway here, then it doesn't matter what G is. And again, the only implication here is in your readout. So if we were doing a cross with genes C and P, you would look at your products. Okay, this is an example from Bateson and Punnett. This is the Punnett square Punnett. What they found is that, you know, I needed both of these to be able to produce my functional product, sure, but if I didn't have gene C, and then it didn't matter if I had P or not, so line 2 and line 4 here, I still didn't produce my product over here. If you were to do a cross with these, you would do the cross normally, Punnett square, whatever, and then you would just have to interpret, okay, this dominant would give me my final product. My dominant C recessive P would give me the intermediate. And anything with a recessive C would give me the precursor. So if this were a dihybrid cross, like we've seen before, I'd have 9 in this first category. I'd have 3 in this third category, like I'd expect out of my 16, but I'd have 3 plus 1. I'd have 4 in this precursor only category. Okay, so seeing those altered statistics is, again, a way to know that there's something non-Mendelian going on. Here's alternate pathways. That one's pretty easy to understand. I'm going to skip past that. Here's inhibition, same kind of idea as the first example, just flipped over on its head where the dominant now stops us. So the only way to get green pigment here is to have recessive for both. So that'd be my 1 out of 16 down here if I did a dihybrid cross of heterozygotes. 1 out of 16 would be green. Here, in this case, would be 3 out of 16 would be yellow. And then here, 3 plus 9, 12 out of my 16 would be white. Okay, so it's just about interpreting your genotypes correctly. Lastly, I want to mention inbreeding. There are some examples in the book that tie to examples of social inbreeding, like royal families having a desire to, maintain the royal bloodline. That often ended up in inbreeding between families. Sometimes geographic, sometimes cultural or religious inbreeding happens for different reasons. Whatever the purpose is, or whatever the driving force is, when we have inbreeding, we increase the frequency of seeing recessive alleles because of the probability of that. So what's shown here is albinism. Albinism is a pretty rare thing in the overall population, but we can see in this family that has extensive inbreeding, this consanguineous mating. You can track this through and try to assign whatever social labels you want to these people, but what we can see is that these offspring are mating back together rather than the family tree spreading out with new genes coming in. We're getting lots of these examples where one branch of the family tree is mating with the other branch. And we see all of this increased probability or increased expression of what should be a rare condition. What we know in general is that inbreeding decreases vigor. So shown on the left, it bothers me that these two pictures are organized differently, but shown on the left are two inbred strains of a plant. This is corn, and the hybrid of those two. And you can see that the plants are growing taller. The ears of corn are bigger. And here's the same thing now with just the fruit. We see the two inbred fruits are smaller. The hybrid is bigger. In general terms, bringing in new alleles is helpful. So if there are some random mutations, those hypomorphic or null alleles, those get sort of masked by wild type alleles of the same genes coming from outside of that original family. We can graph this any which way you want. In those pictures, we just looked at plant height, crop yield. If we look at more and more inbreeding of the species, we see decreased vigor. And inbreeding is measured by this F character, the inbreeding coefficient. I like kind of mentioning this, and there's a short video if you care to watch it, but the father of hybrid rice. You probably are aware that rice is a major staple crop in many parts of the world. It provides a lot of sustenance to a lot of people. Rice is a plant, much like Mendel's peas, that's self-pollinating, and so it was thought to be really challenging or near impossible to create hybrid strains. Um, Mendel did it with his peas. Talked about that a little bit before, but in this case, the work discovered a sterile male strain. And because that strain was sterile, it could not produce the pollen portion of what the plant should produce. That allowed the possibility of mechanical or manual hybridization. Um, and so some hybrid studies were done, and this particular strain, notice the name here, the initials, doubled production. And so this resulted in this person earning the World Food Prize a couple decades ago, and was a huge boon for the rice industry. And again, I'm going to skip the video, but you can watch it if you'd like. What was kind of neat about it is that when we used modern analysis, this was from a paper in 2009, so a handful of years after the strain was developed, and we looked at the transcript. Rice has 12 chromosomes, so here they are lined up. And what they found is that only about 10% of the genes were differently expressed. It's kind of neat. So 90% of the rice is the same, about 10% was different. And those genes that were different mostly were about energy and cellular transport. And so this is sort of an analysis of that. If you want to follow through it, you can. The paper's linked. But the point is that the individual strains were fine, but then by outbreeding them, you got a new strain that was better in a number of ways. So we can analyze inbreeding. Here's an example of a simple pedigree showing, in this case, a single male ancestor that has mated with two different females to produce offspring here. So we would use social terminology here. We'd call these half brother, half sister kind of thing. A and B would be halves. I think, right? Half. I always get turned around on the social stuff. Anyway, they share one common ancestor. And so here's the point. If ancestor C, the common ancestor, if these two alleles were the same and it caused a problem. Now, there's a chance that one of these alleles goes to parent A here and goes to parent B. And then if they mate together, there's a chance that those same alleles go back to this new offspring that's labeled I, so that I gets the same makeup as the original parent C, or the grandparent, I guess. That's called identical by descent. So if that same allele goes to these two different offspring, A and B, and then they recombine together in this new offspring, that would be potentially bad. Identical by descent. The more closely related A and B are to one another, the higher probability there is that that will happen. And we can calculate that. So the inbreeding coefficient F is the probability that two gene copies in an individual are identical by descent. What we're going to do is we're going to look at the pedigree, identify a common ancestor, or if there's more than one, this could be additive, but we'll identify a common ancestor. We'll count the number of individuals in each inbreeding loop, excluding the person we're asking about. And then we'll get a quantity here, 1/2 to that nth power. So let me show you back here. If we were to do that here, we're saying how inbred is I down here. I would say the common ancestor is C. And so I can make a loop just like this. Say I've got 1, 2, 3 individuals in that loop before I get back to where I started. So that would be 3. So the coefficient for individual I would be 1/2 to the third power, which is 1/8. And that's the number you get for breeding of half siblings. There are examples in the book. I don't have the image here, but examples in the book showing you if you have full siblings that mate together, hopefully you can figure out that that would be worse. Half siblings are what we just saw. If you start to look at all sorts of different social structures, there's lots of examples in the book. You can see that the bottom line is the further apart those two individuals are when they mate, the lower the offspring's inbreeding coefficient will be. But you can calculate it with the simple formula F equals 1/2 to the nth power, where n is the number of individuals in the loop, excluding the person you're asking about. If you had more than one common ancestor, you would do that F calculation for each of the common ancestor loops and just add them together. All right, finally, here, going forward, practice. So look at the traditional Mendelism from the previous work. Look at these extensions. Again, use the diagrams in the book as necessary. And remember that when trying to explain something, geneticists usually approach problems with the simplest idea in mind. So this is sometimes called Occam's razor. The simplest explanation is usually the right one. And we're going to start there. So generally speaking, if we were approaching a new trait in the lab, I would say, okay, this is probably controlled by one gene that has two alleles. One of those is dominant. I would test it. If my data from the lab didn't match Mendelian expectations, then I would say, okay, what am I seeing here? Does it look like embryonic lethality, where my ratios are skewed? Does it look like I have an intermediate phenotype, like incomplete dominance? Does it look like I have epistasis, perhaps? And I would design experiments to test those. But we always start simple and expand as needed. And so with that, we're going to wrap up Chapter 4. Please do practice that. And as always, reach out to me as needed if you'd like to chat or have a meeting. We can talk about things. Have a good week and good luck.